So we are going to our next speaker, Suzanne Angeli from Buck Institute. Are you there?
Yes.
Great.
Yeah, I can
we
hear
can
you.
also hear you. Whenever you're ready, please share your screen.
Okay. Hello, everyone. My name is Suzanne Angeli, and I'm a research scientist at the Buck Institute in Novato, California. And today I'm going to talk about two mitochondrial stress responses that I've been interested lately. So I've been interested in understanding how proteins misfold in its relationship to aging. And in particular, I've been interested in the mitochondrial unfolded protein response. So the mitochondrial unfolded protein response occurs when there is a protein misfolding or other mitochondrial dysfunction that occurs inside the cell. So oxidative defects or import failure, mitochondrial mutations, pathogens can all activate a mitochondrial stress response, which much of it has been elucidated, the mechanisms in C. elegans, though it was first discovered in mammalian cells. So unlike the endoplasmic reticulum, UPR, or the heat shock response, it is not as well studied. But what the mechanisms that have been elucidated show that a transcription factor, ATFS1, or in mammals, it's ATF4 or 5, senses the protein misfolding in the mitochondria and translocates into the nucleus to induce a broad transcriptional response, depending on what the insult is. So it's usually chaperones are activated in response to the protein misfolding, but mitochondrial biogenesis or detoxification mechanisms, changes in metabolism, may also be activated, and changes in innate immunity or responses to innate immunity. So I'm going to back up one slide to show that in C. elegans, we see, as in humans, mitochondria decline is observed. So here what I'm showing you is mitochondria that is expressing GFP in the intestines of the worms. And you can see the young worms here have traditionally reticular and healthy looking mitochondria, while the aged worms have a swollen mitochondria that is typical of aging, and it's also correlates with mitochondrial dysfunction. So one way that we study the UPR is by using a transcriptional reporter in which GFP is under the transcriptional control of a mitochondrial chaperone. So here HSP6 is the worm homologue of mitochondrial HSP70. So when the mitochondrial UPR is induced, the worms turn bright green. So here you're seeing this picture is individual worms. The intestines are lit up with GFP, and they are all lined up next to each other. So one way that we induce the mito-UPR is by RNAi of oxidative phosphorylation subunits. So these are multi-complex subunits that if you knock one of the subunits down, it will induce a stoichiometric imbalance. And that is sufficient then to cause protein misfolding and induce the mito-UPR in C. elegans. So here is an example of how I might look at the mito-UPR. So RNAi is typically been induced introduced during the first 3 days of the developmental stages of the worm. And here I've knocked down various subunits from complex 1, complex 3, complex 4, complex 5. And you can see then that this all induces robust mito-UPR as evidenced by the GFP expression. So one thing though, I wanted, and in the past, loss of oxidative phosphorylation subunits during development has been associated with lifespan extension in C. elegans. And so here I'm just showing a survival curve, and you can see in the blue is an example of loss of one of these subunits by RNAi that leads to lifespan extension. I was actually interested, though, to see if we could induce the mito-UPR when the worms were adults. And this is for a few reasons. One is that when you knock down oxidative phosphorylation subunits during development, it often decreases its fitness, evolutionary fitness. So typically the worms are smaller in size. Usually they are sterile. So there are some fitness problems. So I thought if we could try to induce the UPR when the worms are during adults, we could bypass some of these caveats. So looking at these same subunits, the worms were introduced to this RNAi either during development or what I'm calling post-development or adulthood. And you can see that actually most of them knockdowns do not induce a robust mito-UPR, except for this subunit, ATP3. And when I looked at that canonical transcription factor that is known to induce the mito-UPR, I see that during adulthood or post-development, that ATP3 is also regulated by ATFS1, similar to what has been seen in the developmental stages. And HAF1 and UBL5 are cofactors that have been known to work with ATFS1 in the mito-UPR. So it appears to be regulated similarly as the developmental mito-UPR. But when I did a lifespan looking at post-developmental RNAi of ATP3, I saw that instead of having lifespan extension, there is actually a shortening of lifespan, which was surprising and unexpected. So I wanted to really then focus on what was happening during this adult stage that was so different from what we were seeing during development. So I went on to look more closely at what OSCP or ATP3 was doing. So in mammals, the ATP3 is called oligomycin sensitivity conferring protein. So ATP3 is part of complex 5, which is the subunit that makes the complex that makes ATP. And here I'm just showing a schematic of the electron transport chain, and here is the proton rotor in the inner mitochondrial membrane. When protons go through, that helps catalyze the ATPase to catalyze ADP to ATP. And here in the gray, are called peripheral stalk subunits, which are subunits that tether the proton rotor and the ATPase. So when I dug into the literature, what I found was that ATP synthase, which is that the complex that ATP is part of, appears to be involved in the mitochondrial permeability transition pore. So this is a phenomenon that occurs when there is an external stress. And when there are stresses in the cell, there's typically rises in cytosolic calcium. And the mitochondria has mechanisms of taking in that calcium to alleviate some of the cytosolic stress. And but if the stress is too great, what happens is that the mitochondria becomes swollen with calcium, and it must actually then relieve the calcium in a way. And that is by releasing the calcium back out into the cytosol through a pore. And this pore is called the mitochondrial permeability transition pore. And the current models show that ATP synthase is the most likely candidate to be the pore to facilitate this phenomenon. So the mPTP is very relevant to aging and age-related diseases. So it increases with age, and it's also involved in rare and severe mitochondrial disorders, but it's also involved in a host of age-related diseases. So heart attack and stroke or other ischemic reperfusion injuries are known to be in large part, the injury is due to the mPTP occurring. And it's also been shown the mPTP to be elevated in neurodegeneration like Alzheimer's and Parkinson's. So the current, one of the favored current models of how the mPTP functions is that destabilization. So in my case, it appears that loss of OSCP may destabilize the ATP synthase in a way that exposes the potential pore that releases calcium back out into the cytosol. So here, the model I'm showing is that loss of OSCP then exposes the proton rotor, and calcium can then be released out into the cytosol. And there, excuse me, there is evidence that loss of OSCP can actually increase pore formation. So I wanted to see if what I was seeing in C. elegans could recapitulate characteristics of the pore. So the main characteristics are loss of membrane potential, a rise in cytosolic calcium, mitochondrial swelling, and responsiveness to cyclosporin A, which is a well-known mPTP inhibitor. So here I'm looking at the membrane potential using TMRM, and I see that ATP3 does have a loss in membrane potential compared to other subunits. It does have a rise in cytosolic calcium, and it does induce mitochondrial swelling. So it was consistently being shown to be responsive to have pore characteristics. And importantly, the Mito UPR was responsive to cyclosporin A, but not FK506, which is a similar immunosuppressant that is not involved in the mitochondria. And cyclosporin A was then able to rescue many of these phenotypes. So cyclosporin A was then able to rescue the loss in mitochondrial membrane potential and the rise in cytosolic calcium seen with loss of ATP3. And it was also able to rescue that blebbing swollen mitochondrial morphology. And importantly, it was also cyclosporin A was able to rescue the shortened lifespan that we see in the worms and due to loss of ATP3. So in the interest of time, I won't be able to go through some of the other data, but I will just say then that proton rotor, which is also called the C ring, we would hypothesize that loss of that proton rotor would have similar effects as cyclosporin A. And we did see that loss of that proton rotor did actually recapitulate many of the protective effects that we saw with cyclosporin A. And interestingly, it also on its own extends lifespan, which suggesting that the loss of this proton rotor or inhibition of the mPTP can lead to lifespan extension or slow down aging. So the working model is that destabilization of OSCP causes a pore opening, the mPTP, which then may activate ATFS1 to activate the Mito UPR. And I'd just like to thank Gordon Lithgow and Julie Anderson and Anna Folger for her tremendous help as a technician on this project. Thank you.
Thank you so much, Suzanne. Really great talk. We have time for one quick question from the audience here.
Great talk. So I have two questions.
One question?
One question, all right. So Paolo Bernardi has suggested that actually dimers of ATP synthase are what form the pore and not monomeric ATP synthase. Although, I mean, it's still controversial because Johnny Walker has knocked out every single subunit and it persists. So I guess I was wondering how you could apply Bernardi's findings to your own model about loss of OSCP and the dimerization status.
Sure. So I would say it is actually I didn't have time to go through the data, but this is now published. But I did see that my model is consistent with his in that subunits that are also important for the dimer also have similar effects in that they inhibit my phenotype. But I did want to mention the C-ring proton subunit because it was the only subunit I saw that on its own caused lifespan extension. So that's the reason I personally favored the C-ring model. But as you said, it's controversial, and I think actually my findings fit in with the dimerization model too.
Thank you so much. Really great talk. Let's give another round of applause for Suzanne. And if there are more questions, go on Slack and post them, and then Suzanne, you can go on Slack and answer them.
Yes, thank you.
Thank you.